Today what we're going to do is look at categorizing mutations that can happen to DNA and result in a change in amino acid sequence. We're going to look at two types of mutations. The first, depending on what resource you're looking at, sometimes it's called a point mutation versus sometimes it's called a substitution mutation. If you change any nitrogenous base, it has the potential of changing the amino acid. It doesn't necessarily mean that it will. Sometimes a change in one nitrogenous base still codes for the same amino acid. For example, if we look back at our codon wheel sheet, which is the blue sheet, and look at the bottom of there, we see that one mRNA codon doesn't necessarily code for one amino acid. If you look at something like leucine, well, leucine could be the amino acid because of 6 different possible mRNA codons. So one change in one base might not actually result in a huge shift. For example, with the change in amino acid sequence with sickle cell anemia, that's pretty significant. So it depends. I hate using that word, but it really does depend. Well, if we change just one nitrogenous base, let's do the practice. DNA strand of what we call a normal strand of DNA. In genetics, when we talk about something being normal, it's the sequence it's supposed to be. It's not a judgment about whether normal is good or bad. If you look at the change, it's right here. The G22 sequence is one base different. So instead of being an A, it is being a T. Does that change show up in the amino acid sequence?
Yeah.
Yes. You have a different amino acid sequence. Up here, it's supposed to be histidine. But because of that one base difference, that is glutamine now. Everything else is the same. Do you think that this would change the entire amino acid sequence?
No.
No. We have one base difference, so it's just that potential amino acid that can be changed. Do you think that's going to result in a significant genetic disorder? Cool. In the example of what Michael shared, the sickle cell anemia, that one base difference totally changed the shape of the protein. So it could result in a significant disorder, but it is hard to be able to predict that just from an amino acid sequence because you really don't have enough knowledge at this point to predict how the protein is going to fold and be shaped and make a fairly reasonable hypothesis about how that's going to affect the function of that particular protein. The next change is what's called a frame shift. Sometimes this will be referred to as an insertion or a deletion. In a frame shift mutation, you have something that is added or completely deleted from the amino acid sequence. So, what I want you to do is deliberately take out the space here. It does not exist anymore. I'm sorry. Add in a nitrogenous base of your choice. It doesn't matter which one. G, C, A, or T. Add something right there.
Oh, probably because that extra thing. Let's go. C.
I wonder if it was already starting to germinate in the bag. Oops. I'm talking about plants. The original sequence of amino acids was serine, histidine, asparagine, and proline. You were asked to add a nitrogenous base between A and G here. What happened to the amino acid sequence from this point on? Was every amino acid different after serine?
Yeah.
Was one amino acid different? Did you have stops appearing?
You would get stop appearing at AC.
Yeah. So it shifts everything. Because you've added an amino acid, now the next codon after AGA is not GTA, it's something else. And then the third codon completely changes, and then the fourth codon completely changes. If you were to delete the G off of GTA, then the codon would become TAC. So can you see where the name frame shift comes from?
Yeah.
In a frame, we're talking about a codon. It completely shifts one over. Or if you add, then one of those is bumped to the next frame. That is red. And the whole entire chain changes from the point of mutation onwards. Do you think this is a significant mutation?
Yes.
Absolutely it is. This is going to have profound effects on the body. It could mean that the protein that is made is completely dysfunctional. In the case of the cystic fibrosis, the ion channel is not made properly. Therefore, you have a lot of respiratory and digestive issues that result from that. It could even mean death of the individual because a protein is not doing what it's supposed to do in there. Any ideas of what could cause a mutation? So, for example, probably back in your grandparents' days, what kind of safety equipment did they have on the farm when dealing with the pesticides and herbicides? Nothing. You just put your hands in there and scooped out whatever. There was no respirators for dealing with anything. So people came into contact directly with those chemicals. You have to wonder, well, what did those chemicals actually do to the body? Those things could have interfered with DNA replication, and maybe errors could have been introduced. Sometimes it could be ultraviolet light. Have you heard of the ozone layer?
Yeah.
Okay, so the ozone layer is in the stratosphere, way up there, where the planes fly. And it's supposed to block the ultraviolet radiation from coming down to Earth. But in some areas, the ozone is thinner, and it allows ultraviolet radiation through. If somebody's skin is exposed to a lot of ultraviolet radiation, that radiation can actually mutate the DNA and cause melanoma, which is also called skin cancer. So sometimes it's a radiation source. It doesn't have to be radiation from a nuclear power plant. There's many different sources. Sometimes it's random. It just happens for whatever reason. We don't know why. I would like to talk about how is it possible for stuff to be inherited. When Connor was making his presentation on cystic fibrosis, he was starting to talk about how one parent could be a carrier and another parent could be a carrier, and then you would have a 25% chance of having a child with cystic fibrosis. So he was using something called a Punnett square to predict the probability of a child having a genetic disorder. That's starting to get into the realm of genetic statistics, which is actually our next section. And to explore that, we need to look back into the history and look at this fellow named Gregor Mendel, who was actually an Austrian monk. He liked to play with peas. He would do what the bumblebee does. Have any of you ever tickled your pea plants?
No.
No? Or some of the flowers in your garden? You literally take a paintbrush and you tickle the inside of the flower, and then you go to the next flower and you tickle the inside of that flower. And so you are deliberately breeding certain pea plants with each other. And that's what he did. And based on that, he noticed that with things such as color, specifically yellow peas or green peas, whether or not those pea plants were tall or short, whether the pea seeds were wrinkled or smooth, those three things could actually be passed down from one generation to the next. When do you think Gregor Mendel did this work? You think this was something that's really recent?
No. It's probably
I think this was something a long time ago.
Yeah.
Yeah, it was something a long time ago. When was DNA discovered?
1954?
Something like that, yes. And actually, we started calling it DNA. The actual shape of it really wasn't discovered until the 70s when they started using what is called X-ray crystallography. So we have the use of the word factor because we didn't know about genes. We didn't know about DNA back in his time. He recognized from tickling pea plants that things were being passed from one generation of pea plants to another. And he called those characteristics traits factors. But when we hear factors, I want you to think of the word gene. He also recognized that there were different forms of the gene. So, for example, peas are colored. He saw that you could have yellow peas or green peas. So color of pea would be a gene. Green would be one allele. Yellow would be another allele. Alleles are a different form of a gene. He established the law of dominance. And he said that a dominant allele overrules a recessive allele. So let's say yellow is dominant to green. If one parent provided the yellow allele and the second parent provided the green allele, what would the child or baby pea plant be?
Yellow.
It would be yellow because because this is the dominant form of allele and it overrules when we represent alleles, we use letters. It really doesn't matter what letter you use. What does matter is that the capital letter represents the dominant and the lowercase letter represents the recessive. I could have easily chosen the word or the letter T for my previous example. A typical genetics problem will start off with describing the relationship. For example, brown eyes is dominant to blue eyes. In reality, eye color and the genetics of eye color are actually pretty complicated. It makes for a really interesting independent study of somebody else to explore that. So we'll just start off with basic eye color inheritance. I'm going to try to confuse you with how complex it gets. We need to choose a letter. You could have easily chosen E. It doesn't matter which of the 26 letters I choose. You use the same letter for the same gene. So we're going to use B's for eye color. The dominant allele is brown, so that's why we have a capital B there. The recessive is blue, so it gets the little b. So remember that when we inherit chromosomes, one comes from biological mom, one comes from biological dad. So let's say we got one allele, which is going to be the little b, from one parent, and the brown-eyed allele from another parent. What will the child have? Eventually, most babies are born with blue eyes, but then the melanocyte production will kick in and the eyes will change color. If the individual receives a brown-eyed allele from each parent, in order to have blue eyes, they have to get both recessive alleles. So how is it possible to have a blue child if both parents have brown eyes?
Because they both carry the recessive trait, and it gets passed on to their children.
So, and you can have several generations of carriers, and then surprise, blue-eyed child. Which is the case in my family. My grandmother has blue eyes. Nelson family has blue eyes. Which is pretty interesting because she was from a family only one with blue eyes.
So it's a 1 in 8 chance.
We have a couple more words. Genotype and phenotype. I'll let you copy this down and then I'll explain it. When we start talking about a genotype refers to letters that we use to represent the allele. For example, big B, big B, big B, little b, little b, little b. If you were ever asked for a genotype, it's asking for those letters. When you're asked for a phenotype, it's asking you for what characteristic will that individual have. For example, if a child inherits 2 dominant alleles from each parent, or 1 dominant allele from each parent, and is big B, their eye color is going to be brown. So it actually wants to know what the characteristic of the individual. Because the the individual has two dominant alleles. Homo means same or similar.
That's right.
What about the word size? Does that sound familiar at all?
Kind of sounds like Zordon.
What about the word zygote? Do you recall that from science?
I don't know what it's called, but
It's like when the kids play some board game and their house goes to space, and it's like it was a really movie. That was like my favorite movie. Xander.
triggered now. That's my brain.
Okay.
Spiders.
So, when a sperm fertilizes an egg, what results is a zygote?
Left and tail.
That very first cell has formed at conception. It's called the zygote. Then you'll start to see division of that zygote into 2 cells, 4 cells, 16 cells, 32, 64, and so on. So what this is referring to is the fact that if you inherit 2 dominant alleles, you have 2 at the same gene in one side. If you have 1 dominant allele and 1 recessive allele, the characteristic is still the same, brown eyes. But we would describe this as
It's like
being hetero.
cell when
What is
conception when you sperm and eggs. So first
So it's referring to different alleles in one zygote. If the genotype is little b, the characteristic is blue eyes. This is a homozygous condition, but instead of saying the word dominant, we say the word recessive, because both of these are the recessive allele that's been inherited. A few different terms, which will come up again and again, and we'll keep reviewing what those terms mean. Okay, just pause this for a second.